Vectors are biological tools that are needed in gene cloning studies. And in this session, we are going to focus on plasmids as vectors. Rather, in this, we are going to look at what are the characteristics that make plasmid DNA suitable to be considered to be used as a vector in cloning studies. So let us look at the learning outcomes of this session. Plasmid DNA are closed circular extrachromosomal DNA found in many bacteria. It is also found in yeasts. There is a plasmid which is called as 2 micron plasmid that is present in the yeast cells as well. But other than that, mostly you have plasmid DNA found in bacteria. They have an inherent property of being able to replicate autonomously. Plasmid DNA that are used as cloning vectors are small in size, as then it becomes easy to manipulate. Plasmid DNA have two or more genes that can give specific characteristics, which allows the DNA to be identified and selected. To be used as a cloning vector, the plasmid DNA should have restriction sites, which can be sites at which gene of interest can be inserted. Now, therefore, let us consider that you have a bacteria. The bacteria, apart from having its genomic DNA, will also or can also contain, not always, but can also contain what is called as extrachromosomal DNA that are closed circular DNA, and these are the plasmid DNA. So the plasmid DNA can exist as a relaxed DNA, or it can have different conformations based on whether it is supercoiled or nicked. Plasmid DNA can be of different sizes and its copy numbers can also vary. What is also interesting to notice that within a bacteria, you may have more than one type of bacteria being plasmid being present. And this is possible. So it has been observed that about 7 to 8 different types of plasmid DNA can coexist within a bacteria, and that is only when you have these plasmids have compatibility. If plasmids are not compatible, then one or two plasmids will be completely lost from the bacteria. Therefore, compatibility leads to determining how many plasmids can coexist with each other. Now, the question that needs to be asked over here is that what is the role of a plasmid? Is plasmid actually required by the bacteria to survive? So the answer to that is that a bacteria without plasmids can definitely survive, as the primary genes necessary for the metabolism or the growth of the bacteria all lies within the chromosomal DNA. However, the presence of plasmid confers the bacteria with added characteristics, and those added characteristics help the bacteria to carry out certain specialized functions or to be able to grow in certain conditions which are very specific, or they're able to competitively be better than those bacteria that do not have the plasmid. So the role of a plasmid is that it confers certain special characteristics to the bacteria. Say for example, if the plasmid DNA has genes for resistance to a particular antibiotic, then the entire bacteria or the bacterial population will be antibiotic resistant. So let's say for example, you have a media containing antibiotic and you have a bacteria that has a resistance gene in its plasmid that can resist the presence of the antibiotic, then in this media only those bacteria will grow which have the plasmid. So what is interesting to note about over here is this: the plasmid itself is kind of communicating that because the plasmid is present within the bacteria is able to carry out a particular function, in this case resist the antibiotic, and because it can resist the antibiotic, it can grow in the media containing antibiotic. So bacteria that does not have the plasmid, for that bacteria the antibiotic will be uh, a chemical that will stop its growth. But because of the presence of the resistant genes, the bacteria is able to grow in a media that is actually containing a chemical that can stop its growth. So it has a competitive edge over those bacteria that do not have the plasmid DNA. And therefore, this kind of a phenomenon is what is called as positive selection. What does that mean? It means if you have two sets of bacteria, one bacteria without the resistant gene and one bacteria with the resistant gene, obviously of the two bacteria, the one that has the resistant gene will grow. The one that does not have the resistant gene will not grow. So therefore, only those bacteria that have the plasmid will grow over here, which is that it is positively selected over the other. And therefore, such plasmids have a gene that can be considered as a selectable marker. And this is definitely something that makes the plasmid a good candidate to become a cloning vector. Because if the cloning vector carries a gene of your interest and it is present in the host cell, you can select only those host cells that have the selectable marker, which means that the selectable marker is suggesting that the presence of the selectable marker in the bacteria is also essentially trying to say that the cloned DNA is also present. So therefore that becomes a very important phenomenon or very important aspect of plasmid DNA that makes it suitable to be a cloning vector. Now, size again is of prime importance to be considered as a vector. The ideal size is about 10 kb. If you have a plasmid DNA which is lesser than 10 kb, more better. Why is it better? Because the smaller the plasmid DNA it is, the larger size of the DNA insert can be. That means the DNA that can be inserted in the vector can be larger. If the plasmid size itself is 10 kb, then when you add the DNA that has to be cloned, the plasmid will become bulkier, and handling a bulkier plasmid may become difficult. So therefore, any plasmid that is 10 kb and less is a preferred plasmid for acting as a vector, or for being considered as a vector. Now, naturally, of course, there are several different types of plasmids present and or derived from the original plasmid. So E. coli itself, you can see you have col E1 and PUC8. So you can see that its size is 2.1 and 6.4. Therefore, they are good candidates for becoming a vector. But you can also have plasmid sizes as large as 213. Now, these definitely would be difficult to use, but they may have certain characteristics which can be used under certain conditions. So plasmid size can range from 1 kb to 250 kb. Smaller plasmids can be modified and used as vectors more easily than larger plasmids, but larger plasmids may be adapted for very specific uses. So therefore, smaller plasmids are good candidates to act as a vector or to be used as a vector. Another important aspect about plasmid DNA that makes it suitable to function as a vector is its ability to have different copy numbers. Now, what do we mean by copy number? It means that within the bacteria, whether you have just one copy of the plasmid or you have several copies of the plasmid. So, a plasmid DNA generally always has either one or more than one origins of replication. So, because it has the origins of replication, it is an autonomously replicating DNA, which means that irrespective of whether the bacterial gene, that is the chromosomal DNA, or the bacterial genome is multiplying or replicating, irrespective of that, the plasmid DNA can multiply, uh, and so the copy number of a single plasmid within the bacteria can be more than one. Okay, so that is something that is very important to note. Now, smaller plasmids use the host replication machinery to multiply. while larger plasmids may have their own genes for replication. That means they may use certain machinery of the host bacteria, but then there are certain genes that it itself has to ensure that it is getting multiplied. Now, therefore, one another factor that enables the copy number of the plasmid to increase is through a phenomenon of what is called as integrating into the bacterial DNA. So here it is important to note that certain plasmids are non-integrative plasmids, that is, they would be present inside the bacteria, but they would be independently present. They will never integrate into the host bacteria. While in other cases, what you have is that the plasmid DNA can integrate within the bacterial DNA, and as an integrated plasmid, so when it is integrated, it is called as an episome, or the plasmid is called as an episomal plasmid. So an episomal plasmid will multiply when the genomic DNA or the chromosomal DNA is multiplying. So on cell division, with every genome, you would also have the plasmid DNA multiplying. When you have more bacteria being formed, every bacteria will have a copy of the episome. In this case, of course, what you have is that you would have the plasmids being independent. You will have them present separately and in more copy numbers as observed. Here it is dependent on the chromosomal DNA to replicate. Only when the chromosomal DNA replicates, the plasmid DNA will also get replicated. So plasmids that have low copy number are considered to be stringent, and plasmids that have high copy number are considered to be relaxed. Now, if one is looking at cloning, and what cloning means is getting more of the same, then for the plasmids that are more relaxed, for cloning, that those plasmids are more preferred over the ones that are stringent. Now, plasmids can also be conjugative or non-conjugative. What does this mean? Conjugation is a phenomenon that is observed in many bacteria. What conjugation means is, suppose you have a bacteria that has a conjugative plasmid. Now, this conjugative plasmid will have certain genes that will enable forming a connecting bridge between another cell that does not have a plasmid DNA. So, from this bridge that is formed between the two bacteria, the conjugative plasmid is able to transfer a copy of the plasmid into the other cell. Once you have both the plasmids present in the two, you can see how from one cell you have now two cells having the plasmid DNA. These in turn can pass on the plasmid to another uh, uh, bacteria that does not have a plasmid. So eventually, because of conjugative plasmid, an entire population of bacteria can have the plasmid DNA. So this is becoming a very important phenomenon for what is called as horizontal gene transfer. That means the genes can be transferred from one cell to the other cell through a conjugation tube. Now, there are non-conjugative plasmids. That means the plasmids do not have genes that enable them to form a conjugation tube. And since they are not able to form a conjugation tube, the non-conjugative plasmids will not get transferred to a recipient cell. However, if the non-conjugative plasmid is present with a conjugative plasmid, that means you have a conjugative plasmid and you have a non-conjugative plasmid, then along with the conjugative plasmid, the non-conjugative plasmid may be co-transferred. So that is a possibility that is there. But what is essentially important to notice that plasmids can be conjugative or they can be non-conjugative. Now, plasmid DNA is of course classified based on that one specific gene that gives or that confers the host with a characteristic, with a function that is visibly seen or with a function that is uh, is is giving a mark of that particular plasmid. So based on that particular characteristic gene in the plasmid, you can have what is called as fertility plasmids. So fertility plasmids, the word fertility comes from the fact that you can have uh, two bacteria mating with each other. So essentially, you do not have what is called sexual reproduction. But what was observed is because the fertility plasmids, that is the F plasmids, have the TRA genes, TRA genes, the TRA genes enable forming the conjugation tube between two bacterial and therefore you can have DNA from one bacteria getting transferred to the other. So fertility plasmids are example is the F plasmids in E. coli that are basically conjugative. Then you have what is called as resistance plasmids. We've already seen an example earlier. Resistance plasmids are those plasmids that have genes that confer resistance to either one antibiotic or more than one antibiotic. So these are, for example, you have RP4 present in Pseudomonas. So RP4 is a resistance plasmid and it confers resistance to kanamycin, say for example. Then you have another class which is called as the col plasmids. Col plasmids, that is col E1 of the E. coli, for example, have a gene that give rise to colicins. Colicins are chemicals that can actually kill other bacteria. And so these cold plasmids will have a competitive edge over the other bacteria that don't have cold plasmids because the presence of the colicin is going to prevent other bacteria to be present in the same media, and therefore these would confer a competitive edge to the host bacteria. Then you can also have plasmids which are called as the degradative plasmids. So degradative plasmids are those plasmids that have genes that can give rise to a product that can break down a particular polymer or particular recalcitrant product. So for example, you may have the tall plasmids from Pseudomonas putida. These tall plasmids are able to utilize toluene as an energy source. So the tall plasmid has a tall gene which is able to degrade toluene. Similarly, you can have plasmids that can degrade, say for example, crude oil. Okay, so such plasmids that confer the ability to the bacteria to use uncommon carbon sources, then that is what again you can see how the degradative plasmids give a competitive edge for the bacteria that has this plasmid. Then of course you have what is called as virulence plasmids. So virulence plasmids, the best example is that of Ti plasmid. So a Ti plasmid is able to which is present in Agrobacterium tumefaciens, is able to infect a plant cell and cause a disease called the crown gall disease. Since it is causing a disease or a pathological condition in the plant, it is called as virulence plasmid. So the plasmid has genes that is able to cause the disease in the plant. So these are the 5 classes of plasmids based on the main characteristic gene that is present in the plasmid. Now, if you were to look at these characteristic gene for use in cloning studies, the fertility plasmids help in gene transfer. So that is a good function. Resistance plasmids can be used as selectable markers. Colicin plasmids can again be used for selectable markers. Degradative plasmids also as selectable markers, and virulence plasmids as gene transfer. So all of these can be used for as a cloning vector because of its specific characteristic gene. But then it's not just the characteristic gene which is important. You must also consider the size, the copy number, whether it is episomal, non-episomal, all those things will be considered to finally use the plasmid DNA as a cloning vector. Therefore, let us make the conclusions. Plasmid DNA are closed circular DNA that can autonomously replicate to increase their copy number. Plasmid DNA vary in their sizes, with a size of 10 kb being ideal for use as a vector. The plasmid DNA can be episomal or non-episomal, conjugative or non-conjugative. More than one type of plasmid DNA can be present within one cell, and that is only when the plasmid DNA are compatible with each other. Based on their characteristic gene, the plasmids are classified into fertility plasmids, resistance plasmids, cold plasmids, degradative plasmids, and virulence plasmids. Thus, the ability to multiply on its own, the presence of characteristic genes for use as selectable marker or for horizontal gene transfer, the ability to coexist, and variations in size make plasmids suitable to be a vector for gene cloning. Thank you.